Today, I'll be talking about how maternal stress and the maternal microbiome can impact offspring development and social behavior. Now, maternal influence on an offspring's phenotype is an important source of organismal variation that we observe across a variety of taxa. For example, maternal stress can have organizational effects on the brain, which impact the development of offspring and affect offspring phenotype for both males and females. Now, more recently, the maternal microbiome has emerged as a potential novel form of prenatal programming. Now, when we talk about the microbiome, what we're talking about is a complex ecological microbial community that's composed of living microorganisms. And this can include commensal, symbiotic, and pathogenic bacteria, fungi, and archaea. Now, the maternal microbiome, including the gut microbiome, can not only impact the development of their offspring's microbiome, but can also influence the development of offspring physiological systems and also impact behavior. So when we consider the maternal influence on an offspring's phenotype, we not only want to consider how these systems interact independently, but also consider the fact that these systems do in fact interact and may affect offspring phenotype in a unique way. So we want to move forward thinking about how these maternal systems interact to affect male and female offspring development and behavior. The work I'll be talking about today investigated how manipulations to the maternal gut microbiome and maternal stress can interact to affect both offspring gut microbiome diversity and composition, as well as offspring social behavior. I conducted this work in a lab population of Siberian hamsters, which are an excellent non-model system in which to test and ask these questions. Social behaviors like investigation and aggression are essential for interactions with conspecifics. And antibiotics, which have been used to manipulate the microbiome, have been successfully used in Siberian hamsters to not only impact their microbiome, but also to reveal that these can have sex-specific changes in adult behavior. To conduct this study, I exposed pregnant females to one of 4 treatments over the course of their pregnancy: either antibiotics to manipulate the microbiome, a stress-only treatment, the combined treatment of antibiotics and stress, or the control. We then allowed offspring to develop and assessed a variety of characteristics in both female and male offspring. When offspring were 40 days old, we collected a fecal sample, which we used as a proxy to measure their gut microbiome diversity and composition. When offspring were 51 to 56 days old, we conducted a social behavior assay. What we did is we exposed our focal offspring to a novel conspecific for 15 minutes and then assessed the social behaviors displayed towards that intruder. For this first part of the talk, I'll be discussing the results of how manipulations to the maternal gut microbiome and maternal stress affect offspring gut microbiome diversity and composition. The first graph I'll show you here is an assessment of the Shannon Diversity Index of the gut microbial communities in male and female offspring. Now, the Shannon Diversity Index is a commonly used metric to characterize diversity in a community because it accounts for both abundance and evenness of the groups present. So what we found is that the maternal microbiome manipulations did in fact affect the diversity of the gut microbial communities in offspring. Specifically, offspring produced by mothers either exposed to antibiotics in green or the combined treatment in purple had lower diversity, or their microbial communities were less diverse compared to those of offspring produced by control mothers or stress-only mothers. We next assess how maternal manipulations impacted the relative abundance of the phyla detected in the gut microbial samples. So what we'll have here is relative abundance on the y-axis, and then maternal treatment here on the x-axis, with females in the left panel and male offspring in the right panel. And again, what we found is that the maternal microbiome manipulations affected the relative abundance of certain phyla in both male and female offspring. And the phyla that were most impacted by this treatment are indicated here with stars as well as arrows in the main figure. And what we found is that these phyla were significantly reduced in offspring produced by mothers exposed to antibiotics or the combined treatment. And in some cases, they were completely absent. I'll next be talking about how manipulations to the maternal gut microbiome and maternal stress affect offspring social behavior. And I'm going to be focusing on aggression today with assessing individual aggression scores, which is the composition of their attack and chase behavior. What I'm going to show you here on this graph is an assessment of female and male offspring aggression scores. Aggression scores that are more positive or greater are indicative of a more aggressive individual, and individuals with a negative score are individuals that are considered less aggressive. And we again have maternal treatment here on the x-axis. What we found is that there was a sex-specific effect of maternal stress and microbiome manipulations on offspring aggressive behavior. So let's first focus on females. Female offspring produced by stressed mothers displayed high levels of aggression and were significantly more aggressive than other female offspring. In fact, they displayed aggression levels comparable to control males. Now, female offspring produced by mothers exposed to the combined treatment displayed lower levels of aggression and were more similar to that of female control offspring. And it appears that exposure to the maternal microbiome manipulation appeared to alleviate or lessen the effects of maternal stress for females. Now, male offspring, we sort of saw the opposite result. Male offspring produced by stressed mothers displayed levels of aggression more similar to controls, whereas male offspring produced by mothers exposed to the combined treatment displayed slightly higher levels of aggression. So what this work can tell us is that the effects of the maternal environment on offspring development and behavior can be sex-specific, are certainly long-lasting, and can interact to affect behavior and development in sex-specific ways. Female offspring, for example, were more susceptible to maternal stress compared to males. And for female offspring, exposure to the maternal gut microbiome manipulations appeared to lessen the effects of maternal stress. Further, we saw that maternal microbiome manipulations did impact both the diversity and the composition of the gut microbial communities in offspring, and that these manipulations seem to impact the more rare phyla in their offspring. And so with that, I would like to thank everyone that I've worked with in the Demas and Wellman labs, as well as Kate and Luke, as well as and the other undergraduate volunteers who helped with this study. And I would also like to thank our funding sources, NIH and NSF. Because I am transitioning to a new position as assistant professor at Utah Valley University, I'm sorry that I'm unable to be here today to answer questions, but please feel free to email me or find me on Twitter, and I would be happy to talk more about this work with you at a later time. Thank you again for coming to my talk.